Okay, this lecture is for the rest, or for the remainder of chapter 7 that we didn't cover yet. So starting from really DNA repair on. However, I just want to talk about one thing that I had sort of mentioned in class and then I never really clarified. When we were talking about the leading strand, right, and whether this, the polymerase still needs a primer, right, to initiate replication. Remember, we're talking about replication. And that primer actually is an RNA primer, so that means that this figure is sort of inaccurate, right? If this is an RNA primer, obviously there wouldn't be a thymidine here, it would be a uracil. So this is misleading. It's still the same enzyme, the primase, that makes the primer. It just needs to do it once though, right? On the lagging strand, it'll do it multiple times, right? It has to keep putting those primers down to generate those fragments. All right, but it is the same mechanism. It's an RNA primer on the leading strand as well, but it only needs to be there once, right? And then it can go continuously. Okay, let me also now, while we're on this, because I want to make sure I also cover the chapter 7 homework questions and just go over them while we're in here. So the first chapter 7 homework question says, why is synthesis of Okazaki fragments initiated by primase instead of DNA polymerase? Now, the leading strand synthesis, like I was talking about, it's still initiated by, essentially by primase. There still needs to be a primer there because DNA polymerase can never initiate replication de novo, meaning it has to have a primer first. It has to have some small starting template, and then it can add from there. Okay, and so obviously the RNA primers are a little more prominent, and I guess more important when you're talking about the lagging strand. And so that's what that question is trying to get at. But bottom line is, if you answer that saying the DNA polymerase needs to have a primer in order to start replication, that's enough. For the Okazaki fragments particularly, you're going to have to lay down a primer multiple times, right, to generate those Okazaki fragments. And then what happens and maybe we should just go to that picture. And what happens is those RNA primers have to be removed, right? So we have this synthesis occurring for each Okazaki fragment, right? And then eventually those RNA primers get removed, right? And then the gap gets, basically those fragments are joined by ligase, okay? So again, I guess maybe that question is worded a little weirdly, so I just want to make sure I went over that. The next question, because I think we already covered this as well. What is the function of the 3' to 5' exonuclease activity in DNA polymerase? Right? And so again, remember that if we're talking about DNA polymerase in terms of the way it will synthesize DNA, it always has to go, right? 5' to 3'. And so that 3' to 5' exonuclease activity is that proofreading activity, right? And so that means that the DNA polymerase can go backwards if there is a misincorporated base, a wrong base is incorporated, and it can use its exonuclease activity to basically cleave it or cut it out, okay? So that's what that means. So it says, what would be the consequence of mutating this activity of DNA polymerase 3 on the fidelity of replication of E. coli DNA? So what would happen if the DNA polymerase did not have the ability to go back and cut out misincorporated bases, right? So that's essentially what that's asking you. And you can kind of, hopefully we all were able to answer that. And so if we go back, if we kind of move forward here, right, when we were talking about these mutation rates. So, the fact that the mutation rates are so low during replication is a result of that 3' to 5' exonuclease activity, right? So if it wasn't working properly, then that means that there would be many more mistakes made and thus many more mutations, right? So that means that in terms of the fidelity would be decreased and there would be more mistakes, right? So again, we use that term here. We said that you have increased fidelity, right, because of this proofreading activity. If it's not there, then you would decrease the fidelity, increase the mistakes, increase the number of mutations. Okay, the last chapter 7 homework question I will get to when I start covering repair, and I'll come back to that. We didn't talk about double-stranded breaks yet. Okay, so now let me just skip to start talking about the DNA repair part of it. And again, I'll probably skip over some of what we already covered kind of quickly, and then I'll spend a little bit more time or be a little bit more thorough with those things that we didn't cover. We honestly basically covered almost everything. I just wanted to make sure I went over it again, just to be sure, because I know I kind of went through it quickly. Okay, so the first part of the DNA repair section of this, right? I just want to make sure I was on the first slide. Basically talks about types of DNA damage that can occur, right? And remember, we kind of said that, and also remember, you should be filling in the notes that you haven't filled in your review sheet, because there is an assignment to take a picture of it and show me your handwritten notes on the review sheet like we've been doing for the last 2 weeks, okay? So that, and we already started doing this, but just make sure that you have this filled out and fill in anything new that you may have sort of forgotten or realize as you're listening to this again. So remember, DNA changes can occur spontaneously, right? Meaning for no reason, no good reason at all, or from exposure to chemicals or radiation. And so I give you examples of different types of DNA damage and what causes them as we move through, okay? So like I said, I'm going to kind of skip over this quickly because we talked about this already. So a type of spontaneous damage, and you need to know, like I can ask you on the exam, to give me an example of a spontaneous type of damage versus a type of damage that might be caused by a chemical or radiation. Okay, so you need to be able to distinguish between them. So deamination is an example of spontaneous damage here. And you can see, again, it's called deamination because you're losing an amine group, right? So a cytosine can become a uracil. Again, just for no good reason at all, it just happens, okay? And same thing, this adenine can actually change and the amine group be removed. Another type of spontaneous damage is called depurination. So you're basically going to lose the base, okay? So that's what this is showing you here. So where you have basically the backbone, the ribose backbone here, and you're missing the base. All right, so 2 types of repair mechanisms. You can have direct reversal mechanisms, and then also excision repair, meaning that the damaged bases will be removed and then replaced, okay? Again, we went through these already. There are only a few types of damage that can be repaired by direct reversal, okay? One is a pyridamine dimer, which is caused by exposure to UV light. And again, remember that mammalian cells do not have the ability to do this direct reversal and this direct repair of a pyridamine dimer, okay? But other cell types do. Okay, so this is showing you here, exposure to UV light can basically cause this dimer to occur. A dimer is just 2 things coming together, right? There's a bond that forms between them, right? So you can see here, okay, here's your normal 2 adjacent thymines just happen to be in the DNA, and then what happens is they cause this thymine dimer, and so they bond together. And that creates like a blib and a sort of blockage in the DNA. So the direct reversal mechanism to repair UV damaged, right, particularly UV-induced pyridamine dimers, is called photoreactivation. Okay, it directly reverses that formation of that bond, right? It breaks that bond and directly it just fixes and removes that dimer. Okay, so that's what this is showing you again. This is photoreactivation. Also, there are other types of damage that can be caused by chemicals, right? So again, the pyridamine dimer was caused by UV exposure. Here, this is caused by a exposure to a particular chemicals that are alkylating agents, okay? And so they will transfer methyl or ethyl groups to a DNA base. So here's an example here, right, where it's called alkylation, right? You have your guanine, and basically you end up with this O6 methyl guanine, and all it is really this methyl group that gets added on, right? Because it says basically these compounds will transfer methyl or ethyl groups to a DNA base, right? So those reactive compounds or chemicals will cause this to happen, okay? All right, and so then it says that O6 methyl guanine will actually pair with thymine instead of cytosine, right? So it's going to change the binding characteristics or the complementary base pairing that occurs. Okay, so there is a direct reversal enzyme, right, or mechanism that can repair this. And again, this isn't in every single cell, and this is the enzyme that catalyzes that, okay? A long name, but actually makes sense, right? So it's methylguanine, O6 methylguanine methyltransferase, and it will transfer that methyl group, right, off of this methylguanine, because we don't want that there, and basically pull it into its active site. Okay, again, I told you this in class, you don't need to know this mechanism. You just need to know that basically this enzyme is going to remove the methyl group. Okay, again, it's going to directly reverse that chemical reaction, directly remove that methyl group that was added because it was not supposed to be added, and again, changes the complementary base pairing, right? This O6 methylguanine does not bind to cytosine, okay? All right, now those were two examples of a direct reversal, and again, make sure you're writing this in your review sheet here, okay? And I have it listed like that. I have direct reversal of DNA damage, photoreactivation, repair of O6 methylguanine. Those are those two mechanisms. Now the rest of these are different types of excision repair. So this is not going to directly reverse what caused the damage. It's just going to remove the damaged bases, remove the damaged nucleotides or piece of DNA, and then essentially replace it. Okay, so again, we need to know the difference, and you should have the definitions of each one of these explained on your review sheet. Base excision repair, a single damaged base, right? Single damaged bases are recognized and removed. Okay, and so there's an example here that you can actually have uracil arise in DNA sort of by accident, right, in place of thymine, and the cell can actually just remove that remove that uracil and replace it. Okay, and so this is actually now further explaining that process, right? So the enzyme that's involved in that that catalyzes this is called DNA glycosylase, okay? It will cleave the bond between the uracil and the deoxyribose of the DNA, right? To basically free that base, okay? And then allow the new base to be incorporated in, okay? So it's saying, again, if you follow along this, that DNA, I'll show you the picture. I think that's what I did in class. DNA glycosylase will result in the formation of something called an AP site, meaning it's just the backbone of the DNA, the deoxyribose deoxyribose backbone, and no base there, okay? So it's an empty spot, and then you're going to have to fill that in, okay? So let's look at this. So you guys should know the basic sort of mechanism of what's going on here, okay? Again, here's your U. It's not supposed to be there, okay? And so this is basically, it's formed by deamination of cytosine, right? So there's a cytosine there, amine group is removed, and that form uracil. There's other reasons why there might be misincorporated bases as well, okay? So the DNA glycosylase removed just that base. And then now you can see this AP site, meaning there's no base there. There's just the sugar phosphate backbone, okay? AP endonuclease is going to make a cut here, and then basically you're just going to remove that part of that backbone, and then basically you're going to end up adding that back in, okay? So DNA polymerase and ligase will come and add that correct base, right? So you can see there's a series of enzymes involved. I definitely want you to know DNA glycosylase and then understand that DNA polymerase is going to come in and add that correct base. Also know what an AP site is. Okay, so you may want to add some of these notes, AP site, DNA glycosylase, next to base excision repair on your review sheet. Okay. All right, so other excision repair systems. So again, now we're going to give you some examples, some more examples of different types of DNA damage, right? So bulky groups that can be added to the DNA by carcinogens, and also UV pyridamine dimers like we talked about before, okay? So if you can't directly reverse a pyridamine dimer, you have to use this type of excision repair to get rid of it, okay? So here's another example of another type of DNA damage that can happen by radiation and chemicals, okay? So here's showing you guanine, and if you come in contact with a carcinogen, it can cause this bulky group to be added in, okay? And so nucleotide excision repair will remove damaged bases as part of a larger fragment, right? It says an oligonucleotide. It's not just going to remove one base, okay? And so this Exonuclease is a complex that's going to directly excise that oligonucleotide. Excise meaning cut out, right? It says helicase is required to unwind the DNA, right? Remember, we talked about all these things with DNA replication. Again, you're still going to have whatever that resulting gap is. It's always going to be DNA polymerase and ligase that's going to come in, fill it in, and join it together. Okay, so this is showing you, again, going back to that thymine dimer. Okay, again, if the cell does not have the ability to do photoreactivation, this is the way it needs to get rid of that thymine dimer. All right, so again, you can see that it's basically going to cleave out this entire fragment here, right? This one, it's one strand, okay? And then polymerase will come in, add in the correct bases via complementary base pairing. Okay, these repair genes, right, have actually been identified in studies of certain diseases, okay? So there is a disorder where you, in fact, have extreme sensitivity to UV radiation. And as a result of that, you have multiple skin cancers, right? And basically, the genetic target of this disease is a gene that is going to code for enzymes involved in nucleotide excision repair, right? So if your cells don't have that ability to fix UV-induced damage, right, you're going to obviously end up with a lot more damage and thus obviously more skin cancers from there, okay? And so you can use these cells, right, these XP cells actually as an experimental system to really look at the function of DNA repair genes and to identify DNA repair genes. Okay, so it says we use cloning to identify repair genes that are mutated in individuals with XP, like I mentioned, but also in other diseases as well. Okay, and actually these proteins that they identified are closely related to other proteins that are encoded by yeast. Okay, so they're called these RAD genes. So basically, when we're talking about these molecular cloning, we're basically relating human DNA repair genes with these yeast DNA repair genes, okay? And that these are essentially the same genes. So the same genes responsible for, if we want to be specific here, nucleotide excision repair in yeast are the same type of genes. They're highly conserved in humans as well. Okay, another type of excision repair, transcription coupled repair. So this is nucleotide excision repair, except that it's happening during transcription. So if the problem wasn't fixed before transcription was going to occur, transcription starts. However, if that thymine dimer is there, the transcriptional machinery is not going to be able to get past it. It's not going to be able to transcribe the DNA where the thymine dimer is or past that. Okay, so it's super important that it actually gets fixed. So RNA polymerase can actually stall, and then there's specific proteins that will recognize the damage and then recruit proteins in to repair what's wrong. Okay. All right, so that's for transcription coupled. Now, this specific next few slides here is basically giving some specifics about mismatch repair. Okay, so there is a system, okay, and so I have this as a separate category here. So if you again look at your review sheet, right, we talked a bit about base excision repair, nucleotide excision repair, transcription coupled repair, and then mismatch repair. Okay, so this is think of this as a separate system for that is going to specifically look for mismatches. Okay, so it'll scan newly replicated DNA. So we said the DNA polymerase has the ability to fix mistakes. However, it misses stuff, right? And so there's another sort of check right after DNA replication is finished to check to see if there are any mismatches that have occurred. Okay, and so in E. coli, that newly synthesized DNA, okay, is not yet modified by methylation, meaning the cell is going to be able to recognize what was the template strand versus the newly synthesized strand. And so if you want to take a second to think about it and think about why that's important, right? If the cell's going to fix something that's misincorporated, it needs to know which one is the right one, right? It needs to know which one was part of the template and then that the other one was just synthesized during that round of replication, okay? And so one way that it can do that is that what will happen eventually is that the DNA will become methylated. However, right after replication, the newly synthesized strand does not have methyl groups added onto it. Okay, so again, the cell can recognize the template strand, the parental strand, because of that. Okay, there are specific proteins that are involved in this process. Okay, so mut-S, mut-L, mut-H. I'm going to show you a picture. So it says the protein mut-S will recognize the mismatch. So it's the one that points it out and identifies it. And then it will form a complex with these other proteins here. Then there's this mut-H endonuclease will cleave that unmethylated DNA strand, okay, and basically unwind the DNA and repair it, okay? So this is basically just showing you, so I want you to understand the general mechanism here. Again, this is showing you here that the old strand, the template strand, is methylated. The new strand is not, okay? And so again, that's what allows the cell to know what's old versus new. And so obviously it's gonna fix this mismatch base, not this one. This is the correct one, this is the wrong one. Okay, so these are some 3 proteins, enzymes that are involved in this. Okay, and so one will identify it. Okay, we go back here. We said the mut-S sort of recognizes that mismatch, but then recruits all of these other ones here and basically cleaves out, you know, the area of the DNA that the mismatch was in and replaces it. Okay, so in terms of what's happening and how it's removed is just like we talked about before when we were talking about nucleotide excision repair. Here, though, you have other proteins specifically involved in identifying that mismatch. And again, this is particularly for mismatches that occur during replication, and they're detected right after replication is finished. And that was in E. coli, okay? In eukaryotes, there appears to be, or the ability to distinguish between the template strand and the newly synthesized strand doesn't have to do with the methylation like it does in E. coli, but that apparently there are single strand breaks in that newly replicated DNA, right, that haven't maybe been completely sealed up, and that's how they can specify the strand, the template versus the newly synthesized, okay? And there are eukaryotic homologs of those same proteins we just mentioned before in E. coli. And again, remember, homologs just means it's a, you know, a gene that basically has the exact same function in a different species. Okay, so this is what this is showing you here, right? That you can identify sort of these mismatched cases just because of these small nicks that are believed to be in the newly synthesized strand. And then again, homolog, so that's why they're called MLH, because it's saying it's human, right? Compared to the E. coli version of these proteins, the same idea. One will come and recognize it, recruit other proteins, other enzymes to the area, which will cleave out the mismatched, you know, section of DNA, and then it will be, you know, filled back in, and then basically we can continue replication. Okay, so again, this is right after kind of one part of it's been replicated. Again, this is sort of another level of, you know, repair, another type of repair mechanism to get rid of any mistakes that might have happened during replication. We said DNA polymerase alone can kind of back up and fix its mistakes, but that's sort of immediate. This is, again, another level of repair. Okay, and again, the why should anyone care? So mutations in those human homologs of those mut-S and mut-L that we just talked about, right, that are part of this mismatch repair system, cause a specific type of colon cancer, right? So there's this inherited type of colon cancer, and it is one of the most common types of inherited diseases, and it's one of those cancers that really is genetic, right? Obviously, all cancers have a genetic component, but this one, it has been, you know, specific genes that lead to it, or mutations in specific genes, have been identified, okay? So again, defects in these mismatch repair genes result in a high frequency of mutations, okay? And basically, it increases the likelihood that, you know, of cancer developing because of these mutations that occur. Okay, so again, that's the sort of why do you care? So we gave examples of, you know, that pigment disorder, that XP, that's caused by defects in repair genes. Here, particularly in these mismatch repair genes, if you have mutations in them, it leads to this particular type of colon cancer. All right. So another type of DNA, and so this is kind of goes along with this. It's called, and it's kind of it's on your review sheet here, translesion DNA synthesis. So again, make sure you wrote in something for Miss Repair. Translesion DNA synthesis. This is if there's a protamine dimer or other type of lesion that's going to basically block the DNA polymerase. It actually has the ability to sort of replicate across the site of damage. All right, sorry, a little pause there. So let's just talk about this translesion DNA synthesis. So if all this or some of these types of DNA damage that we've been talking about do not get fixed, right? The cell has a way sometimes of getting past or gets past that DNA damage, right? So it says specialized polymerases can actually replicate across the site of DNA damage because, again, if it wasn't able to replicate across, then basically, you know, that's it. Nothing's going to happen, right? And then it'll sort of give it one more chance to be repaired after replication has occurred. So these are, again, some specialized DNA polymerases, right, that we just mentioned here, right? We said specialized DNA polymerases. So again, this is yet another level of sort of, or another type of repair mechanism that's going to give a sort of another level of accuracy or another level sort of security, right, to have the correct DNA basis. So polymerase 5 was discovered actually in 1999, so that's getting closer to your guys' birth dates, in E. coli, and actually it is a specific polymerase that's actually induced in response to UV radiation, and it can synthesize a new DNA strands across a thymine dimer, right? So the UV radiation would induce that thymine dimer, and this specific polymerase will actually be activated to replicate across. Polymerases 2 and 4 are also induced by DNA damage. So bottom line is this is just giving you a sort of brief description of, you know, examples of this, of how there are these specific types of polymerases that have been identified. Okay, so eukaryotic cells also have specialized polymerases for translesion synthesis. These are particularly in E. coli. All right, so this is just showing you a picture of this. Again, here's your thymine dimer, here's your normal polymerase. And so if it reaches this thymine dimer, it's not going to be able to synthesize across. And so this special polymerase needs to come in to basically synthesize the DNA across that thymine dimer. Okay, and then once it gets past it, then other polymerase will come back in to finish the process, and then right after, it can then be fixed. Okay, all right, so that's that translesion DNA synthesis. Again, make sure you write notes in there, particularly understanding that there are different types of DNA polymerases or specialized DNA polymerases that can actually replicate across the damage. And in particular, we gave an example of a thymine dimer, but it could be another type of damage as well. All right, the next one is repair of double-stranded breaks. So everything we've been talking about so far, right? Anything that's happened is basically on just one strand, and the cell still has the template strand to know what's supposed to be incorporated there. Okay, so as you might imagine, if you lose or you have a problem on both strands, it's going to be the cell doesn't know what the correct base sequence is supposed to be. Okay, so it says they're particularly dangerous because the continuity of the DNA molecule is disrupted, but these happen, so they can occur naturally or can be caused by radiation or chemicals. Okay, so there is a specific mechanism. Okay, so again, repair of double-stranded breaks, you should be writing in recombinational repair. Okay, so recombinational repair is a mechanism to repair double-stranded breaks. Okay, and if you guys go to look at your chapter 7 homework question, it says describe 2 mechanisms the cell uses to repair double-stranded breaks in its DNA. One is recombinational repair. Okay, so double-strand breaks are repaired by simply rejoining the broken ends of a single DNA molecule. Super simple, just joins it. Okay, but there's a lot of errors involved with this because there's basically deletions of bases around that site of damage. Okay, so again, super simple. Here's your radiation or something else that might have caused this double strand break. and then basically you just glue them together, okay? And you lose some basis at the end where it gets glued together. All right, so that's the first one. The second one is, the second repair mechanism for double-stranded base is homologous recombination, basically, okay? And so it can use the information on the other undamaged chromosome, right? Remember, we have 2 pairs of chromosomes, right? So it doesn't have the template strand on that same, chromosome on that same piece of DNA, but it can use the information on the homologous chromosome. Okay, so it says this only occurs following DNA replication when the newly replicated sister chromatids remain associated. Okay, and so that's what this is showing you here. So it doesn't need to use the, it can't use the information on the template because it's all gone, right? But it can use the information on this undamaged DNA because these are perfectly lined up, right? In terms of what DNA sequence is here, it's the same. And so you can see how these strands sort of intertwine with each other. And basically you have synthesis occurring sort of using this as a template here, then here using this as a template, and it gets filled in. Okay, and then kind of goes back. And so you can it's kind of showing you can see where the darker brown and the lighter brown is, a sort of mixture of the sort of undamaged chromatid, or yeah, I guess it's a little, all right, whatever. Anyway, of the undamaged chromosome, we'll just say that, okay? So, so that's the way it's going to work. It's not just going to be able to just use that again, not going to be able to use the template strand because that information has been lost as well. Okay, so homologous recombination, meaning it's going to use an undamaged, if you go back here, right? DNA sequences on an undamaged chromosome. Okay, so again, obviously it has to be the same chromosome. It can't just be a random chromosome because then we put wrong sequence in there. But again, as long as we're talking about the homologous chromosome, right, then you can use that information. So the cell's pretty smart, right? It has a way of trying to get that correct information in there where that double-stranded break is. All right, so 2 mechanism, it just ligates it or glues it together. The other one is it uses homologous recombination to basically fill in that gap with the correct DNA sequence, with the correct nucleotides that should be there. So you're not going to lose nucleotides like you would in just gluing it together. Okay, now there are some key proteins here, and again, these will relate back again to cancer. And so that's why I have this last thing here as repair mechanisms and the relationships to cancer. This is what I'm referring to. And so again, take some notes here. So one of the key proteins in that homologous recombination, right? So that doesn't just happen randomly. We're leaving out a whole bunch of details in terms of the mechanism here. There are specific proteins that are involved in this, okay? And so one of those key proteins is called REC-A in E. coli or RAD51 in eukaryotes. And again, remember, these are homologous proteins, okay? They're conserved, right? They have the same function. REC-A has the same function as RAD51. But REC-A is in E. coli, RAD51 is in eukaryotes. So they'll bind to the DNA and form this, and then bind to the double-stranded DNA and basically catalyze the exchange of these strands, right? So see how this is kind of happening, the exchange of these strands here? It's basically catalyzed by proteins and enzymes, okay? And again, one of the key ones is REC-A in E. coli and RAD51. These 2 proteins, though they're in different species, right, in different cell types, they have a similar function, okay? And so this is actually just showing you electron micrograph where you can see that REC-A sort of catalyzing this reaction, okay? And this is just showing you that RAD51. Actually, let me just see what that is exactly pointing out because it's kind of, 1 second. Just want to see if the figure caption gives us any more information on what that's specifically, maybe what that's showing there. No, not really. Yeah. So let me see, 1 second. Where is that? Yeah. I mean, so in fact, it's actually removed from the new version of the textbook probably because it doesn't really show you much. So anyway, it's just a picture, an electron micrograph showing you those two proteins. Okay, so again, I'm not giving you any specifics about the mechanism. The idea is if you go back to this picture here, you basically can add, you know, Rec-A or Rad51 kind of in here at the sort of site of where this is happening, and they're going to basically orchestrate the infiltration of these strands and allow this homologous recombination to occur to basically fix that. And so they're the main players in this. If they are damaged, this is not going to be able to happen, okay? All right, so a little further with that. So one of the genes that's responsible for inherited form of breast cancer, right? We've probably all heard of the BRCA gene. It actually encodes a protein that will recruit Rad51 to basically, it says the single-stranded DNA at the double-strand break. So what the heck are they referring to? If we go back here, you know, basically you have this double-strand break. It's going to basically recruit the protein here. Okay, that's all it's referring to. So it's the BRCA gene itself is not Rad51. However, it encodes for a protein that will work with Rad51. Okay, so like I said, Rad51 is not the only protein involved in that process. Okay, but bottom line is that BRCA gene is involved in this homologous repair mechanism that we just talked about, okay, of double-stranded breaks. And again, if you have defects in this type of DNA repair, it can lead specifically to the development of breast cancer. So again, another type of cancer that is associated with a specific gene responsible for DNA repair. So for the BRCA gene, specifically associated with this double-strand repair, the homologous recombination double-strand repair, okay? We also talked about colon cancer, and then we also talked about other types of disease, pigment disease, that XP, okay? So make sure you can make the connection between mutations in DNA repair genes and cancer, right? And why defects in these repair mechanisms or mutations in these genes might lead to the formation of cancer, okay? And so again, that's that last thing there. Okay, this last part on DNA rearrangements, you're going to put an X through it on your review sheet. We're not going to cover it. It's not going to be on the exam. So I'll know if you listen to the lecture or not. So you should have an X through that on your review sheet. All right, guys, so that's it for chapter 7.